Imagine trying to crack this massive, heavy iron safe. You've got your stethoscope against the dial, you're spinning the wheel, and you finally click that first tumbler into place.
Oh, the best feeling, right?
Exactly. But then the moment you do, the internal mechanisms of the safe physically rearrange themselves. The lock you were just picking, it just no longer exists.
That is honestly a brilliant way to visualize what an oncologist is up against every single day.
Right, because that shifting safe is essentially a cancerous tumor when we attack it with just one drug. For a long time, monotherapy was, our main strategy.
Yeah, we'd find a powerful compound, deliver it to the patient, and really just hope it was the silver bullet. But cancer is terrifyingly adaptive.
It really is. So when you hammer a tumor with a single treatment, you're placing this immense evolutionary pressure on it, right?
You are. The cancer cells that happen to have a natural resistance, they survive, they multiply, and suddenly you've triggered a large drug resistance.
The tumor literally mutates around your weapon.
Exactly. And to try and force that single drug to keep working, doctors often have to push the dosage incredibly high.
Which leads to those just debilitating side effects. I mean, the toxicity hits the healthy cells just as hard.
Right, which limits the therapeutic effect and dramatically heightens the risk of treatment failure. You're basically poisoning the patient to catch a rapidly mutating target.
But our entire approach shifted back in January 2014, right? That was a massive watershed moment.
It was huge. The FDA approved the first combination therapy specifically targeting melanoma patients, specifically those who had very specific genetic markers, the BRAF V600E or V600K mutations.
So we stopped trying to pick one lock and started attacking the whole mechanism at once. We combined the drugs.
Precisely. And since that moment, over 50 non-fixed dosage combinations have been sanctioned. The driving force behind all of this is a concept called drug synergy.
Right. It's not just taking two drugs and getting the sum of their parts. It's this biological math where 1 + 1 suddenly equals 3.
Synergy is the multiplier effect. When drugs are truly synergistic, they amplify each other, so you can lower the actual physical dosage.
Which spares the patient from those brutal side effects, right?
Exactly. While simultaneously trapping the tumor. You hit its primary growth pathway with drug A, and you block its escape route with drug B. You box the tumor into a corner it literally cannot mutate its way out of.
Let's unpack this, because our mission for today's deep dive is exploring a massive, deeply complex review paper on how AI, specifically deep learning, is shortcutting this whole agonizing process.
Yeah, finding these combinations the old-fashioned way is essentially impossible.
Because doing it manually in a lab would take forever, right?
Utterly impossible, both mathematically and financially. I mean, we have thousands of approved chemical compounds and countless variations of cancer cell lines.
Right.
If you rely on wet lab scientists to exhaustively test every conceivable drug pair at every possible concentration,
A physical petri dish.
you'd be running experiments for centuries. We need a shortcut to predict the multiplier effect before we ever touch a test tube.
But wait, before we can train a neural network to find synergy, we have to teach the computer what synergy actually looks like. How do scientists even prove two drugs are working together rather than just acting independently?
That is the foundational hurdle. In a laboratory setting, what we call in vitro, we rely on 4 classic mathematical models to score whether synergy is actually happening.
Okay, let's go through them.
The first baseline is called low additivity.
Low on additivity. Okay, I'm trying to picture this. How does low define teamwork?
Well, low operates on a very specific assumption, which is that a drug cannot interact with itself.
Right, obviously.
So imagine you have a dose of drug A that achieves a certain effect. If you swap out half of drug A with a mathematically equivalent dose of drug B, the overall effect on the tumor shouldn't change at all.
Ah, okay. So low looks for synergy by calculating an additive effect based purely on that strict dose equivalence.
Exactly.
So it's looking at the raw volume of the active ingredients. But wait, what if the drugs don't work the same way at all? What if they attack completely different parts of the cell?
That's the perfect question. And it's why we move to the second model, which is Bliss independence. Bliss approaches the problem through the lens of pure probability.
Probability, like rolling dice.
Sort of. It assumes the two drugs are acting entirely independently, hitting completely different biological pathways. Under Bliss, synergy only exists if the combined tumor destruction exceeds the mathematical probability of their independent effects happening at the same time.
So it's like flipping 2 coins. You can calculate the exact odds of getting 2 heads.
Yeah.
If you suddenly get way more double heads than the math predicts, you know the coins are rigged. or I guess in this case, synergistic.
That captures the spirit of it perfectly, but Bliss has a known blind spot. If the two drugs actually do happen to hit the exact same biological pathway, the Bliss model gets totally confused.
Really? Why is that?
Because it assumed they were acting independently, so it will overestimate the synergy, giving you a false positive.
Oh, I see. Which means we need, like, a tiebreaker.
Exactly. And that brings us to the highest single-agent model, or HSA. This is arguably the simplest metric to grasp.
Okay, lay on me.
HSA defines synergy strictly if the combination of the two drugs performs better than the maximum inhibition achieved by the single best drug in that pair alone.
Let me test an analogy here on you. HSA is like a corporate relay race.
Okay.
If you and I are on a team, our performance is only considered synergistic if our combined time is faster than my absolute best solo time. Like, it doesn't matter what you bring to the table unless the team beats the superstar's personal best.
I like that a lot. But stay with that relay race scenario for a second. What if both you and your partner are objectively very slow runners on your own?
Hey, I resent that.
But your baton handoff is so incredibly flawless that you actually win the race.
I get it. Our solo times were terrible, so the benchmark to beat was incredibly low.
Precisely. HSA can severely underestimate synergy if both drugs are weak as single agents but transformative together.
So all three of these models have flaws.
Right. To solve all these blind spots, researchers rely heavily on a fourth model called ZIP, or Zero Interaction Potency.
ZIP, okay, what makes it different?
ZIP is a hybrid. It integrates the dose equivalence of Lowy and the probability of Bliss, but instead of looking at single points of data, it looks at the entire dose-response surface.
A surface? You mean like a 3D topographic map?
Exactly. Imagine a landscape where the X and Y axes are the escalating bases of the two drugs, and the peaks and valleys represent how many cancer cells die. ZIP maps that entire terrain.
Wow, okay, so it's incredibly robust against the flaws of the simpler models.
It is. So we take these four models, Lowy, Bliss, HSA, and ZIP, and from those landscapes, we extract hard numbers to feed the AI.
And the paper mentions the combo score and the S-score, right?
Yes. The combo score is a direct derivation. It measures the raw difference between the observed combination response in the lab and the expected additive effect, usually calculating that expectation using Bliss or Low.
So a higher combo score just means you have stronger synergy.
Right. And then there's the S-score, which is much more surgical. It measures the percentage of tumor inhibition that exceeds expectations at a very specific, crucial dosage point.
Which point is that?
It's called the IC50.
IC50. What does that stand for?
It stands for the half maximal inhibitory concentration. It is the exact dose required to inhibit 50% of the cancer cell's growth.
Literally half the battle.
Literally. Interestingly, if you look back at older machine learning methods, like a famous random forest model built by a researcher named Gavert, they often relied on a slightly different metric called GI50.
What's GI50?
It's the concentration to inhibit 50% of cell growth. It was simpler for early algorithms, but modern deep learning requires the nuance of the S-score at the IC50 threshold.
Okay, so just to pause and make sure we're on the same page here, let's do a quick recap. We started with a massive problem. Monotherapy fails because tumors adapt.
Right.
Synergy fixes this by amplifying efficacy and lowering toxic doses. And we measure that synergy in Petri dishes using 4 mathematical lenses. We've got Low for equivalent doses, Bliss for independent probability, HSA for beating the superstar drug, and ZIP to map the entire 3D landscape.
You nailed it. And we extract the combo score and the S-score from those.
Perfect. So think about your own workflow for a second if you're listening right now. If you're tackling a big collaborative project at work, which of those models reflects your style? Are you like a Bliss independent worker who stays in your lane, or an HSA where you only want a partner if they can help you beat your personal best?
It's a fun thought experiment. But, transferring those concepts from a human brain into a neural network is a monumental translation problem. You can't just insert a physical chemical and a living cancer cell into a laptop.
No, obviously not. We have to turn physical reality into raw data. The paper refers to this translation phase as feature representation. So how do we teach an algorithm to actually read a pharmaceutical drug?
We use a few brilliant techniques. The foundational one is called SMILES, Simplified Molecular Input Line Entry System.
SMILES. I love when scientists come up with a great acronym.
It really is great. SMILES takes a complex, three-dimensional molecular structure and flattens it into a compact, single-line text string. It uses letters and symbols to denote atoms and bonds.
Okay, so if the drug is a physical building, The smile string is basically the written police report describing it, like it's a 2-story brick house with a red door.
That's a super helpful way to think about it. But a written report loses the spatial relationship, right? If you want to know exactly how the chimney connects to the roof, a text string isn't enough. So AI architects also use molecular graphs.
So that's like the police sketch instead of the written report.
Much better than a sketch, actually. It's a mathematical network. In a molecular graph, the drug is mapped out like a subway system.
A subway system. How does that work?
Every single atom becomes a node, or a station, and the chemical bonds between them become the edges or the train tracks. This preserves the 3D topology of the drug perfectly.
Oh, wow. So the AI is reading the text description and mapping the subway system simultaneously.
Exactly. And then it gets even more granular by generating fingerprints. The algorithm essentially creates a checklist of structural puzzle pieces.
What kind of pieces?
Well, there are substructure key-based fingerprints like MACCS. MACCS acts like a literal checklist, scanning the drug to see if it contains predefined structural fragments. Does it have a specific carbon ring? Check.
Yeah. And the paper also mentioned circular fingerprints, right?
Yes, like Morgan or extended connectivity fingerprints. Instead of just looking for isolated puzzle pieces, these look at the neighborhood.
The neighborhood.
Yeah, they pick a specific atom and analyze the sphere of information around it within a defined radius. It tells the AI how the atoms are physically influencing each other locally.
So we have the drug completely digitized. Text, subway maps, and fingerprints. But Synergy takes 2 to tango. We also have to translate the cancer cell into data. I mean, you can't just tell an AI, hey, this is lung cancer.
Definitely not. Not if you want accurate predictions. The AI encodes cancer cells using staggering arrays of multi-omics genomic data.
Multi-omics. That sounds intense. What does that actually include?
It looks at gene expression profiles, so which genes are turned on or off. It looks at copy number variations, structural mutations, DNA methylation, and gene dependency. It builds a complete, high-resolution molecular portrait of the tumor's specific vulnerabilities.
That is, I mean, to process molecular graphs and genomic portraits of that size, the AI must need an absolutely colossal amount of fuel to train on.
Oh, the datasets driving this field are awe-inspiring. In the early days, we had the O'Neill dataset. It was a pioneer, mapping out 583 drug combinations across 38 drugs.
Honestly, that sounds like a lot of lab work, but for deep learning, 583 examples is basically a light snack.
Exactly, it's nothing. So the U.S. National Cancer Institute stepped in and created NCI Almanac. The scale of this data set is staggering. They systematically mapped over 304,000 samples.
Wait, 304,000? That's insane.
Yeah, using 104 FDA-approved drugs across 60 different human cancer cell lines.
That is a data factory, not a laboratory. But the researchers didn't stop there. They opened it up to the world with crowdsourcing, didn't they?
They did. They built DrugComb, which is this mammoth, constantly updating repository. By mid-2021, DrugComb had ingested over 739,000 combinations across thousands of drugs and cell lines.
Almost 1 million combinations.
And beyond sheer volume, we're seeing qualitative leaps with portals like Syntax Profiler.
Oh, right. What makes Syntax Profiler different from the massive ones like DrugComb?
It acknowledges a really harsh clinical reality. A highly synergistic drug combination is completely useless if it kills the patient.
Yeah, that's a pretty big caveat.
Right. So Syntax Profiler uniquely evaluates synergy, efficacy, and toxicity all simultaneously. It trains the AI to find the needle in the haystack that is both lethal to the tumor and entirely safe for the human.
Okay, time for another quick recap of our journey through the digital looking glass here. So we know we have to translate physical biology into data.
Yep.
Drugs become 1D smiles text strings, 3D molecular graphs, and structural fingerprints like MACCS or Morgan. Cancer cells are mapped out using these massive genomic arrays. And all this gets fed into colossal engines like NCI Almanac and the crowdsourced giant DrugComb.
It's bottom.
So imagine you are that AI for a moment. If you're listening to this and you were trying to understand a completely unknown chemical, would you prefer to read its smiles text report, or would you want to trace its molecular subway map?
It's interesting because the truly advanced AI architects realize they don't actually have to choose. They use both. And that brings us to the actual construction of these neural networks.
Right, the architecture. The paper divides them into 2 basically warring philosophies, right?
Yeah.
Single-task learning and multitask learning.
Exactly.
Let's pull apart single-task learning first. As the label implies, these models have one singular obsession: predict drug synergy. They literally don't care about anything else. And from what I read, they generally organize themselves into either branch structures or graph structures.
That's the main structural divide, yes. Branch architectures operate like parallel lanes on a highway. You have one dedicated lane processing the features of the drugs and a completely separate lane processing the genomic data of the cancer cell line.
Okay.
Models with names like Deep Synergy, Deep DDS, and DFFDDS use these parallel lanes to great effect.
But if they are in completely separate lanes, how do they ever combine the information to find synergy?
Well, they eventually merged the lanes at the very end of the network to make the final prediction. Some models, like MGAEDC, go a step further and actively construct specific drug combination features based on interaction graphs before the merge.
Right. But the true standout in the single task category was a model called SynergyX.
Yes, SynergyX is fascinating.
The paper spent a lot of time on it. It uses a mechanism called mutual attention. How does that actually work?
So mutual attention throws away the idea of isolated parallel lanes. Instead of processing the drug and the cell line totally independently, it dynamically captures cross-modal interactions.
Cross-modal interactions.
Yeah, as the AI is processing the data, it calculates how much the cancer cell line should be paying attention to specific features of the drug and simultaneously how the drug should prioritize vulnerabilities in the cell line.
Oh, wow. So instead of two separate analyses merging at the very end, the data streams are constantly having a conversation with each other. They're updating their understanding based on what the other side is doing.
It's a highly complex conversation, yes.
Yeah.
But that's still just the branch structure philosophy. The alternative is the graph structure. Graph models look at the data not as parallel lanes, but as one massive, tangled, interconnected web.
And the prime example the paper gave here was hypergraph synergy. Now, the prefix hyper usually implies something is turned up to 11. What is a hypergraph compared to a normal molecular graph?
Good question. So in a normal graph, like that subway map we discussed earlier, a line or an edge connects exactly two stations. Drug A connects to drug B.
Makes sense.
But biological systems don't work in isolated pairs. A hypergraph allows a single edge to connect multiple nodes simultaneously. It can draw a single mathematical relationship connecting a drug, another drug, and a cancer cell line all at once.
That is wild. It basically upgrades the data from a one-on-one text thread to a massive group chat. It captures the multi-way relationships that standard graphs just physically can't draw.
Precisely. It allows the AI to see the whole biological ecosystem at once. But here's the catch: single task models, no matter how clever their architecture, are facing really stiff competition from the multitask learning models.
And this is where the paper gets incredibly compelling to me. Multitask AIs have side hustles. Their main job is to predict synergy, but they deliberately cross-train on auxiliary tasks. Why force the AI to do extra work?
Because biology is deeply intertwined. One popular auxiliary task is drug sensitivity. Models like MARSI and MTL Synergy are trained to predict how sensitive a cancer cell is to just a single drug, while simultaneously trying to predict the synergy of a combination.
Oh, so by forcing the AI to deeply understand the baseline sensitivity first, it becomes much sharper at predicting the multiplier effect of synergy.
Exactly. It's building foundational knowledge.
And I assume the other major side hustle is toxicity.
Yes. Predicting drug interactions, or DDI, and adverse side effects, models like Muthine and Deep Trace Synergy are forced to predict toxicity alongside synergy.
Which grounds the AI in the physiological reality of the human body.
Right, ensuring the combinations it finds aren't just the theoretically lethal to cancer, but practically safe for patients.
So the researchers essentially set up a cage match. They took the massive drug comb data set and benchmarked all these different models against each other. And they decided that any combination hitting a low Z-score over 10 was considered highly reliable synergy. So who won the showdown? The laser-focused single-task models or the cross-training multitask models?
The results were highly illuminating. On pure accuracy, the single-task models SynergyX and MDN-NSYN actually took the crown.
Wait, really? The side hustle models lost. All that cross-training didn't pay off.
They lost, but the researchers discovered it was actually an issue of data quality, not the architecture itself. SynergyX and MDN-NSYN won because they construct unbelievably detailed multimodal features.
Okay.
For example, SynergyX uses those substructure partition fingerprints we talked about. It's simply feeding on much richer, higher resolution data.
Better fuel equals better performance. Makes total sense.
But the researchers didn't stop there. They performed what's called an ablation study on the multicast models like MARSI and Muthine.
What's an ablation study?
They went into the code and deliberately turned off the auxiliary cross-training tasks. They basically forced them to operate as single-task models.
Oh, wow. And what happened?
Their ability to predict synergy noticeably deteriorated. The ablation study definitively proved that even without the ultra-premium data feeding Synergy X, cross-training on a side hustle made the multitask models punch far above their weight class.
Okay, let's distill this architectural battle down for a second. Single-task AI zeroes in purely on synergy, utilizing branch structures like Synergy X with its dynamic mutual attention conversation, or graph structures that use hypergraphs to connect multiple nodes at once.
Yep.
Meanwhile, multitask AI takes a different route, cross-training on side hustles like drug sensitivity or toxicity. And while premium data extraction wins the day on pure accuracy, auxiliary cross-training undeniably elevates a model's underlying intelligence.
That's a perfect summary.
It really makes you think about human skill acquisition, like cross-training undeniably works. If you're listening, have you ever picked up an entirely unrelated hobby? Maybe something creative, and suddenly found it made you much sharper at your rigid analytical day job.
It builds a totally different kind of cognitive elasticity. And frankly, we are going to need every ounce of elasticity we can get because this brings us to a rather harsh reality check.
Yeah. The AI models we've discussed are miraculous, but they're obviously not currently curing every cancer in our clinics.
No, they aren't. There is always a gap between the theoretical computer model and the physical hospital bed.
And the best way to predict the future of a technology is to look at what it currently cannot do. Right now, this field is battling a massive issue they call the concentration gap.
The concentration gap is really the final frontier right now. Current AI models, even the incredible ones we just discussed, are mostly binary.
Binary in what way?
They are fantastic at predicting if two drugs are synergistic, but they fundamentally fail at telling us the specific concentrations needed to achieve that biological magic.
I was trying to think of an analogy for this. It's like having an audio engineer listen to a raw track and tell you, hey, the bass and the vocals are going to sound incredible together.
Right.
That's great information. But they refuse to tell you where to put the volume sliders on the mixing board to actually make it sound good. You still have to guess and check.
And in pharmacology, guessing and checking takes months of painstaking wet lab experiments to find the perfect dosage ratio. A synergistic pair can become antagonistic, literally actively harming each other's effects if the ratio is off by just a few micrograms.
That is terrifying. So if we want AI to hand us fully actionable medical blueprints, future neural networks must be able to output exact drug concentration.
Exactly. And to achieve that level of precision, the AI is going to need some entirely new side hustles to cross-train on.
The review paper outlines where the field is heading with this, right? What are the new tasks?
Future multitask models will need to master disease target association. This forces the AI to predict interactions between drugs and entirely novel, previously undiscovered disease pathways.
So it stops the AI from just looking at known cancer mechanisms and forces it to map the broader biology.
Yes. And the second major auxiliary task is something called DTA, or drug target affinity.
DTA, why is that one so crucial?
DTA predicts the exact binding affinities between drugs and their cellular targets. It's not just asking the binary question, does this molecule bind to this protein? It asks precisely how tightly does it bind.
It measures the grip strength.
Yes, exactly. By understanding the exact grip strength, the AI can catch incredibly subtle structural differences between very similar compounds. This level of granular physical understanding is what will finally allow the algorithm to recommend highly rational, exact concentration ratios.
Okay, so we know where we are going. Let's look back at where we started. We began with the heartbreaking statistic of millions of lives impacted by cancer and the frustrating evolutionary walls of monotherapy. We explored how synergy breaks those walls by making 1 + 1 = 3. We mapped out the traditional MAFLO, BLISS, HSA, and ZAP. We watched physical chemicals get flattened into smile strings and blown up into multi-omics genomic arrays, all to feed the massive dataset beasts like drug cone.
It's a massive pipeline.
It really is. And then we watched architectures battle it out, proving that cross-training and mutual attention are the keys to the future.
It is a profound shift in how we do science. We are no longer just observing biology, we are mathematically simulating its deepest interactions.
And that leads to a deeply provocative question to end on. Right now, billions of dollars in the most advanced computational architectures on Earth are strictly focused on destroying mutated cancer cells.
True, but step back and look at the underlying mechanism we have built. We are training deep learning networks to perfectly map the synergistic effects of multiple chemical compounds on human biology.
So if we can perfect this multitask AI to pinpoint exact concentrations and affinities, could this exact same technology eventually be flipped?
It's a fascinating thought. I mean, instead of solely deploying this AI to fight disease, what happens when we use it to map positive synergy?
Right. Could we use these hypergraph networks to design personalized vitality cocktails, precise mathematically validated combinations of compounds engineered to synergistically optimize your specific immune profile?
To slow down cellular aging and maybe permanently shift the paradigm of medicine from fixing what is broken to systematically upgrading human biology.
A daily vitality cocktail designed for your specific genomic array by a multitask neural network. It sounds like science fiction, but given the roadmap we just walked through, it feels like a very plausible future.
It really does.
Thank you for taking this deep dive with us. Keep questioning the incredible hidden math running the world around you, and we'll see you next time.